Good evening and welcome to this Retina UK webinar. This is one of a series of webinars we will be hosting and we'll be delivering at least one on a different topic each month. We are really pleased to have with us today Avril Watson, a PhD candidate of stem cell biology in Professor Majlinda Lako's lab at Newcastle University. Avril is also a scientist at Newcells Biotech Ltd. She is funded through the Marie Curie Action project Stargardt, which focuses on diagnosis, treatment and modelling of Stargardt disease. Her research is focused on modelling Stargardt disease with mini-retinas generated from patients with late-onset Stargardt disease. So whilst Avril is giving her presentation evening, we'll be collecting your questions. So if you'd like to ask a question, please type it in the Q&A section at the bottom of your screens, and we will endeavour to answer as many of those questions as we can at the end. If we're not able to get to your question today, it will be followed up over the next couple of weeks. So thank you again for joining us, and without further ado, I'm delighted to welcome Avril.
I'm just going to share the screen. Okay, hopefully you can all see that. So thanks again for joining me this evening. I'm really excited to be here and glad that you were able to join. Today I'm going to talk about Mini-Retina, which is really a fantastic tool that we have nowadays to study inherited retinal degenerations and really any condition that can affect the back of the eye or the retina. So I've titled this talk A Tool to Unlock the Future of Restored Vision in Retinal Blindness, which I think this tool really is important for. So like Matt introduced, my name's Avril Watson. I'm a final year PhD student here at Newcastle University. And I'm working on Stargardt disease, which is an inherited retinal degeneration. So I thought I'd give a little bit of a background to me. So if it isn't already noticeable from my accent, I'm originally Irish. I also have very characteristic Irish features, the red hair, the freckles and the green eyes. And I lived in Dublin for the first 23 years of my life before making the move over to Newcastle here in the north of England. So I've been here for maybe about 3 years now, and I think I've settled in really well. I think the Irish and the Geordies get on quite well. And I am enjoying life here at the moment, so it's nice to be here. Aside from kind of my background, I also do really enjoy music. So you'll often find me in the lab, walking around with headphones on, listening to music, singing in my tissue culture when no one's listening, and I do like to play guitar in my own free time as well. I love dogs, like many people. Once I see a dog in the street, I'm always the first over to try and talk to it and make it be my friend. And like during the lockdown, many people, I became a plant mom. So my apartment back home is currently like a mini jungle, but I wouldn't have it any other way. So that's just history of my personality. But in terms of retinal research, I've actually always been interested in retinal research. I moved to Newcastle about 3 years ago to continue my studies with my PhD. So I'm currently in Professor Majlinda Lako's group here in Newcastle University. And Linda is really well known in the field of inherited retinopathies, particularly for her research with mini-retinas and generating these mini-retinas from many people who have inherited retinopathies. Separate to this, she's also really well known for studying conditions at the front of the eye. So she's been involved in the process of generating corneal transplants. And a lot of the work that we do here in this research lab will be described as translational. So that means that we'll find something that we can maybe one day turn into a therapy and help people gain back their sight. So this whole process of inherited retinopathies has always been very interesting to me. And in my undergraduate and my master's degree, I also studied different forms of retinopathy. So in my master's in Trinity College Dublin, I had the opportunity to work with Professor Sarah Doyle in her lab there. So she worked in the School of Medicine. And again, a lot of her research was focused on retinopathies. And in this project, I got to work on age-related macular degenerations. And I used mouse models of these conditions to try and understand how the immune system can actually influence different retinal disorders. So that was really interesting and kind of moved my research more towards using cell models for research. Before that, And this is probably the first instance of where I had exposure to inherited retinopathies, and it kind of kickstarted my interest was when I worked with Professor Jane Farrer for a short project in my undergraduate year at university. And Jane is really well known for all of her work in inherited retinopathy. She does a lot of work on retinitis pigmentosa in particular, but one project that is still ongoing in that lab 5 years later is a project called Target 5000, which basically aims to sequence all estimated 5,000 individuals in Ireland that live with inherited sight loss, to sequence their genes and try and find out what particular mutation they have that is causative of their condition. And this really, really fascinated me. I really enjoyed being able to see the patient aspect of the research and, ultimately diagnosing patients through their genetics. I thought it was a really fantastic process, and it really kickstarted my interest. So from this, I knew I wanted to have my career in inherited retinal diseases, so much so that I left a very cushy lifestyle back home in Dublin, where my family and my boyfriend lived, to come here and continue my studies. So hopefully I'll be able to illustrate how exciting this research is and how it's moving in the right direction, and that one day we'll have even more therapies available on the market for people with inherited sight loss. So if I'm going to talk about mini-retinas, I kind of need to go back to basics here and describe what the retina is. And this really is kind of a difficult thing to explain to people who, don't really study the retina, because it is a very complex tissue. But this tissue, it's located at the back of the eye, and its most important role is obviously the process of vision. So from a researcher's perspective, I would arguably say that it's probably one of the most beautiful tissues to have the opportunity to work with. And this is because within the retina, there's this very unique and very pretty patterning of cells that we're able to actually see through various different processes that we can carry out here in the lab. But I think probably the best way for me to describe this patterning of cells is to compare it to cake. So cake is obviously well known. I think we all enjoy cake. But for the purpose of this, I wanted to imagine a multi-layer cake. So this cake has a vanilla layer, a lemon layer, a chocolate layer, you name it, any layer you want. And between each layer, we also have a nice dollop of buttercream. And the retina is very similar to this because there's also these layers within the retina. And like each flavor of a cake is, instantly recognizable by your taste, each layer of the retina is full of these cells that are very easily recognizable by their role in the whole process of vision. And like the buttercream, there's also these spaces between the layers that actually allow the cells to make connections with each other and to talk to one another and pass on information that they've gained from maybe one side of the retina to the other side of the retina. So I like to imagine that process a little bit like a game of Chinese whispers, where maybe one cell will get the first message and it'll be passed along a chain of different cells until it reaches the last cell, where it can actually then be recognized and be processed, or maybe the message can be spoken out loud. So like vision, the light comes in from the front of the eye, it's passed along these chains of cells, and then once it hits the last cell, which is actually in your brain, you can just see what's there in front of you without even thinking of it. So it really is a fascinating process. The fact that this just occurs without even thinking of it is really fascinating. So we try and mimic this with Mini-Retina. But before I talk about the Mini-Retina, I want to talk about dysfunction within the retina and why that's important for when we study Mini-Retina. So to talk about dysfunction, I need to introduce you to three main cell types. So I'm going to try and not bore you with too many details, but probably the most important cells in the retina are the photoreceptors. And these cells are the ones that can actually detect light, and they can convert that light into a signal that's then read by your brain and is processed to a visual image. So we have two different types of photoreceptors. There's rod photoreceptors and there's cone photoreceptor cells. So with the rods, they're usually really important for low light vision. So they are really sensitive. They don't need an awful lot of light to work, but they don't have very high acuity. So you wouldn't be able to see sharp details with these. And they're usually the types of cells that are active at nighttime for night vision. But the opposite of these are the cone photoreceptor cells, and these aren't very sensitive, so they actually need an awful lot of light to work. So with that, they can actually recognize different colors, and they can combine different colors to make even new colors. So with the cone photoreceptor cells, these are very, very, they have high sensitivity in the terms that they can recognize very fine details. So we would use a lot of cone photoreceptors for maybe reading a book or watching the television or driving a car maybe. So they're very important cells. And then another kind of layer of cells that's really important in the retina is the retinal pigment epithelium. And these have many, many roles in the retina, but probably one of their most important roles is that they support these photoreceptor cells. So they do form very, very tight layers between each other where that they can communicate, and the retinal pigment epithelium is responsible for providing nutrients and food to the photoreceptor cells, but also clearing away any waste that's produced during the process of vision. So when visual blindness occurs, it's usually a consequence of a defect in one of these cells. So when these cells stop being so healthy, vision becomes compromised pretty much. So the reason why I'm telling you this is because if we want to be able to develop effective therapies that can slow or stop the whole process of blindness, we need to find a way of keeping these different cell types alive and keeping them alive for as long as possible. So now to talk about mini-retina. So mini-retina is basically retina that's produced in a normal human body. Mini-retina in particular is developing retina. So it would be the type of retina that you would see in a baby that's forming in its mother's uterus. So it has all of the same patterning of cells. This layer, the cake is basically still there. But the only thing is we're missing this retinal pigment epithelium that supports the photoreceptor cells. But it's still a really, really important model because it has all of the other neurons that are present in the retina. So that means that we can actually study a lot of diseases using these mini-retinas. So you might want to ask, why are mini-retinas so fascinating and why are they so important in our studies of inherited retinopathies? And basically, there's three main things that highlight the importance of mini-retinas in inherited retinal research. So the first thing is for disease modeling. So we know that there's many, many, many different forms of inherited retinopathies. There's many different genes that cause inherited retinopathies. I think to date, there's over 300 different genes that are actually implicated in the development of an inherited retinal degeneration. And obviously, we don't know absolutely everything yet. We're still learning. Every day, we're finding new facts between different inherited retinopathies. So with this model, we're actually able to find out even more details about a disease that we might already know an awful lot about from other models, such as animal models, or maybe just from human experiences itself. But with these organoids, we're able to actually take cells from patients that might have a very rare phenotype or a rare symptom of this disease, and we're able to model it in a lab and actually see what's going on inside the eye, because obviously we can't take someone's eye and see what's going on in there. So this is kind of like a proxy. We're able to model this patient's condition, but we can make many little mini retinas or mini eyeballs, and we can study things in our own time and be able to find out things that we wouldn't be able to find out from just looking at someone's eye through a fundoscopy microscope or anything like that. So another really important benefit of using these mini retinas is that we're able to actually perform a process known as drug screening. So with drug screening, you basically have a large panel of drugs that might show benefit to any particular condition. Big pharma usually do a lot of drug screening, but because they have such large panels, they might not actually be testing these drugs against cells that are, unique to whatever condition that they're looking at. So for example, in the retina, if you're using a drug for retinitis pigmentosa, you want to be able to test this drug on a model that is specific to the retina, because there's no point checking it in, say, kidney cells or liver cells, because they're not the same type of cells. We need like a neuronal cell model, and the mini-retina actually serves as a platform for this. So it actually helps us bring new drugs to the table, drugs that might actually potentially cure a lot of conditions. So in big pharma, a lot of these drugs are just thrown out the window because they'll show toxic effects, but they're not being tested in the correct cell types. So we serve this platform for testing drugs to see if there is any toxic effects of the photoreceptors or if there's any toxic effects of the RPE, stuff like that. So it's a really useful model for testing new drugs that could eventually make it to market one day. And then another kind of study that we would do is developmental studies. So we're able to actually track what stage of development in the human being that our organoids develop to. So in our lab, we do a process called single cell sequencing, and it's basically where we can take each individual cell of the retina that we've developed in the lab here with stem cells, and we can compare it against cells that have been taken from fetal retina or adult retina, and we can compare pretty much cell by cell how similar they are to each other, and we can get an idea of how old our organoids or our mini retinas are here in culture. And that's really important because we can actually see whether individual conditions occur during development or if they are actually occurring much later in life, because a lot of conditions you only get maybe the symptoms in your 20s, maybe later on, but we can actually see signatures of dysfunction during the developing retina, which is really interesting because we wouldn't be able to see that without the use of these mini retinas. So in our lab, we do study quite a few different conditions. We work on a particular form of retinitis pigmentosa. We also have some age-related macular degeneration projects ongoing. And we also study other forms of macular dystrophy. And I myself am working on Stargardt disease. And another interesting one, we work on retinoblastoma as well, which isn't really, in some cases it's inherited, but a lot of the time it's somatic as well, as in like you just gain a mutation and then you can actually develop the condition. And using mini-retinas, we've actually learned a lot about each of these individual conditions. In the case of retinoblastoma, this is a childhood cancer. And using the organoids, we were actually able to pinpoint which cell type that the cancer actually starts in. So it's really interesting to be able to do that. And we've also been able to do drug screening and test new chemotherapeutic agents on these organoids to see if we can actually stop the cancer from forming in that cell type. So it is really exciting research that hopefully will make a difference one day. For Stargardt disease, we use Mini-Retina as a tool to see what the effects of the disease are. And in particularly, I'm working on a small subset of Stargardt disease cases that tend to have a late onset of disease symptoms. So instead of having a classical onset of maybe 10 to 20 years, and these patients actually don't get any symptoms until they're about 40 years of age. So we're using mini-retinas to kind of try and figure out why, because if we can figure out how these patients have such a delayed onset, maybe we can try and target that and see if we can actually delay the onset of disease in other classical cases and let people have their vision for much longer. So basically mini-retina are really useful for many, many conditions, and hopefully they will make a difference one day. But just to give an idea of an overall process for one condition, I'm going to talk about a form of retinitis pigmentosa type 12, and this particular condition is caused by mutations in PRPF31. So I mentioned earlier on that our organoids, they don't contain retinal pigment epithelial cells, but we can actually develop these as well from stem cells alongside the retinal organoids. So we can actually have two different models of the retina growing at the same time, and we can compare differences between each of these that are occurring. So this is what we did for this study. It was basically, we generated many retinal pigment epithelial cell lines from patients that had this condition, and we also generated some mini retinas. And we grew these for 230 days. They take a really, really long time to grow. If you imagine a human fetus takes a full 9 months to grow, it's actually not too different for the eyeballs either. So we basically did that. And we were able to see very, very disease-specific symptoms in our patient cell lines when we compared them with healthy control cell lines. So in the mini-retina, we were able to see shorter cilia. So cilia are basically these hair-like structures that are really, really important for the health of the photoreceptor cell. And without these, the photoreceptor cell can't absorb light, and the process of vision can't occur. In the supporting tissues, the retinal pigment epithelial cells, there was defective waste clearance. So any of this waste that the photoreceptors were producing, the retinal pigment epithelium were not able to deal with that. They weren't able to break it down. And a result of that was actually that you got a buildup of all of this waste molecules, which ultimately ends up killing off the retinal pigment epithelial cells. And we see this in patients that have retinitis pigmentosa. They ultimately lose their photoreceptor cells because the retinal pigment epithelium underneath becomes compromised and it actually begins dying off. So then this isn't around anymore to feed the cells that live above it. So then ultimately everything ends up dying, and that's why you end up losing your vision. So it was really fascinating to be able to see this in cell models that we developed in the lab. And one way that we were actually able to stop this is we tried with a drug called rapamycin. And rapamycin was actually able to stop this whole process of buildup of these waste molecules, and the waste pathways were able to be activated again. And this completely removed the disease symptoms in these cell models. So it's really promising that these types of drugs, if brought further and further tested, could actually stop disease progression in actual patients. So it's really exciting research, and it's really fascinating that you can see the translation of the work that we do in the lab to someday make it into patients and help with their symptoms of blindness. So talking about actually going into patients, I'm showing a picture here on the screen of a young guy. He's 24 and he's sitting in hospital here with his mum. His name is Jake Turnant, and he is actually from County Durham, which is quite near where I am at the moment. And he was the first patient in the UK to actually receive a gene therapy called Luxturna for one of his conditions that caused inherited blindness. And this picture, it was taken during the pandemic. You can see them both wearing face masks. And I find it really inspiring. But one quote that he said was that the last year for a lot of people was actually a very dark and miserable year. But for me, it was easily the best year of my life. And that's because he was treated with this gene therapy that was actually able to restore his vision. So for the first time in 24 years, he was able to actually see again. So it is really, really fascinating. Jake in particular, he was gradually losing his vision since he was born, and it was due to a condition that pretty much wiped out one of these proteins, a piece of the machinery that was essential for vision. And so to fix this problem, scientists came up with a way of packaging the instructions to make this missing piece of machinery into a little virus of all sorts. Basically added this virus to the back of his eyeball with an injection, and then this virus was able to infect the cells there and release these instructions for making that protein that was missing, or the piece of machinery that was missing. So with this missing piece of machinery, then the whole process could occur again, and Jake was able to see again. He was able to do this process of vision, and his vision was restored. So it's a really promising story and really exciting. And it really does highlight the fact that retinal research is probably one of the most exciting fields at the moment, because it is rapidly developing. Every day there's new things coming out, there's new therapies on the markets, new things in clinical trials. So hopefully we do see a lot more success stories in the future, and many more people do gain their vision back. So of course, there is a lot of challenges with this. The main challenges really is that the cells of the retina, they don't regenerate. It's not like your skin cells where they're constantly replenishing themselves, like your hair continues to grow. With the retina, once the cells die, they're gone. Unfortunately, they can't come back. So when we're thinking of developing a therapy, we need to really focus on keeping those cells alive. We need to also focus on getting there before the cells die. So that's kind of the first challenge. The second challenge that we need to encounter is the fact that Many people in the UK and in the world, in fact, they don't actually have a genetic diagnosis of their condition. So many people will go to the doctor will look at them, and based on their symptoms, they'll say, oh, you have retinitis pigmentosa, or you have Leber's congenital amaurosis, you have Stargardt's disease. But very few people actually undergo genetic sequencing and they get their mutation that is actually causative of their condition. And this is really, really important, because if there is new therapies that get developed, and there's always these clinical trials, but to be eligible to actually go for a clinical trial, you need to actually have a genetic diagnosis of your condition so that they can make sure that you're an appropriate candidate for this therapy. So if there is anyone who, is thinking about genetic screening, I would really, really recommend going forward for it, because this piece of information is very, very crucial for not only your diagnosis and your prognosis of how the condition will, develop over time, but it's also really important in case any therapeutic options do come up that you're eligible for, that you might want to enlist in. Another challenge that we face is there's an awful lot of variability in inherited retinopathies, and I'm sure you're all quite aware, even within families that have the same condition, the symptoms are not always the same. Some people always get the symptoms earlier, some people get them later. Some people might not get them at all. So it's crazy. Like, even though you might have the genetics to say that you'll have this condition, you might never develop it in your life. So there is a lot of variability, and this kind of needs to be addressed before making kind of a one-size-fits-all therapy. It's also really difficult because making a therapy is a very, very expensive process. And to do this, they need to have many people with the same condition so that they can actually invest the money in. If there's one person with one condition and one person with the other condition, it's not going to be easy to decide which patient will get the therapy. So they're trying to find ways of maybe treating symptoms that affect everyone. So types of treatments that might, increase the health of photoreceptors in all conditions would be an amazing therapy, because it would help so many people. So that's kind of the main 3 challenges. But there's also difficulties in the strategies. So like, sometimes the strategies are just too difficult and you can't actually do the same types of treatments that you might be able to do for other conditions. So like in this condition that I just mentioned with Jake, his particular condition suited the type of therapy that was generated. So basically it was just a missing piece of the puzzle. They were able to give him the missing piece of the puzzle, and that was it. His vision was restored. When a lot of other cases, it's more complicated. Sometimes the missing piece of the puzzle is far too big to be able to put inside a virus. So they need to come up with new approaches to be able to actually, deliver the instructions to cells to make these proteins, but we're just not there yet. But everyday advances are being made. So, it's not to say that this will always be a problem. It's just one that we have to encounter in our research at the moment. Finally, another challenge is cell replacement. So, I mentioned that we use stem cells to make mini-retinas. So you might be asking, why can't we use stem cells to, make these photoreceptors to replace the unhealthy ones that are in eyes, or maybe to make retinal pigment epithelium that we could put in the eye, we could transplant it in there. And yeah, that's definitely an approach that people are investigating. And I think in the future, it actually will hopefully be a process. But at the moment, there is still issues with rejection. So you might have heard from people who might have had kidney transplants or other types of tissue transplants. They don't always get accepted in the body. So we need to find a way that if we're going to transplant these cells into the eye, that we know that they're going to stay there and it's not a waste of your time or a waste of resources, that ultimately it'll work out for you and you don't have to undergo this process of pretty much having surgery on your eye. So they're the main challenges, but I don't want to leave it on a bad note. So I do want to say there is a lot of hope in this field. Like I said, it's rapidly advancing. Every day, new advances are being made. And it is a really exciting research field to be in. New therapies are being conceptualized in labs, and new conclusions are being made, and it's only a matter of time before more therapies are on the market to prevent inherited blindness. And finally, I do want to commend those with blinding conditions for their bravery and their resilience, because you play an enormous role in this retinal research. Like, it's very easy for me to talk about this, but I myself have never experienced retinal blindness. So, it is really, really inspiring that people do want to be involved in this, and they're, very open to sharing their stories. And I really appreciate that as a retinal researcher. So Yeah, that's pretty much it from me. I hope I didn't bore you guys too much, and I hope I was able to pass on how exciting retinal research is. And if you do have any questions, please feel free to ask them. Or if you're a little bit shy, my email is also on the screen and you can send me an email at any time. And I've also included a website for the research that we do in Linda's lab if you're interested in finding out more. So thanks very much.
Avril, that's absolutely fantastic. Thank you so much. That was really, really interesting. So for you people looking at home, watching at home, please do leave your questions in the Q&A section. So the website that Avril was just referring to is www.retinalstemcellresearch.co.uk. We'll share that information in the email we send as a follow-up. So Avril, my first question to you is, how do you grow a mini retina? What's the process that you go through?
Oh, so it's a long process. Pretty much what we'll do is we'll first take some skin cells from the patient that we want to make the mini retina from. So this first process, we take a biopsy. So it can either be a blood biopsy or it can be a skin biopsy. And we'll basically reprogram these cells. That's the process that it's called. So we'll take back the fact that they're a skin cell or a blood cell, and we'll bring them back to the state where they're a stem cell. So stem cell is essentially a cell that can become any other cell in the body. It's where we all have originated from. So it's really going back to the beginning. And with this, then we can, put it in a dish or in a flask in the lab, and we can add different food and different supplements and chemicals that'll pretty much tell that cell, I want you to be a retina cell. And we'll do that, and we culture them in suspension. So that what that means is they'll form these little balls, and they float in the media, and just almost like magic, they can somehow become retina that just self-patterns itself. So we actually don't have to do much. We just have to feed them every day. So we're basically mums and dads here for maybe 6 to 8 months. We feed them 3 times a week. And at the end, we have these mini retinas. They have photoreceptors that are somewhat functional. And they're not really on par with adult photoreceptors, but they're very, very similar to maybe the type of photoreceptors that you would find in a 30-week-old fetus. So that's really the process. But like I said, it's very time consuming, very long-term cultures. We really do take care of them with all the efforts that we have. But yeah, at the end of the process, they form really nice mini retinas.
Excellent. I'm assuming you don't kind of feed it a Happy Meal 3 times a week.
No. Carefully formulated media. It gets added. It's basically just liquid food. It's like feeding your flowers with plant food whenever you buy them from the supermarket. Something similar to this.
Excellent. That's fantastic. We've had a comment rather than a question. So comment is, thank you so much, Avril. That was so interesting and definitely gives me hope that one day there may be a treatment, or even better, a cure for RP. Certainly something that we are all hoping for.
Yeah.
So what area of kind of treatments do you think is the most promising at the moment for the next round of treatments, cures, whatever it is we want to call them?
So it really depends. I mean, for me, I would read up an awful lot about Stargardt disease. So I would probably know more potential cures for Stargardt disease. So for this Stargardt disease, at the moment, there's a new therapeutic strategy for a very specific type of mutation that can actually cause what's called a splicing defect. So this is pretty much when you have your genes that are expressed in your body, and sometimes they're not expressed in the correct way due to these splicing defects. So basically they found a new therapy that's able to better modulate or manage the type of splicing that will take place in a cell. So they're actually able to say, no, you're not supposed to do this, you're supposed to do this. And then that will ultimately stop this splicing, this event happening. And then ultimately you get the correct version of that gene being made in the body, and then everything that follows, then the cells remain healthy. Oh, that's probably a bit complicated. Even me, it's a little bit hard to get my head wrapped around that type of technology. But there's many different therapeutics for many different conditions, but I would probably know the most for Stargardt disease.
Excellent, excellent. That's fantastic. So in terms of the different therapies, I mean, you've mentioned stem cells, so I know there's lots of research and things going into that. You know, what other potential therapies are out there for people with these conditions?
So at the moment, a lot of it is just kind of trying to keep the cells alive. So a lot of it is like advice. So for age-related macular degeneration, for example, they tell people to not have vitamin A because vitamin A is, they tell people to supplement with vitamin A. People with age-related macular degeneration should have more vitamin A because it actually helps the cells stay alive for a little bit longer. So most of the therapeutics at the moment aren't really classical therapeutics. They're more like supplements to try and delay the progression of disease. For the moment, most of the main therapy that's come out is Luxturna, which is what I described Jake is receiving. That's probably the most successful to date. It's the only one that's been FDA approved. But there's various ones in clinical trial that will hopefully be deemed safe to be actually treated in patients. And then we can have something that will actually, stop progression rather than delaying it long term.
Excellent. So what can people with inherited retinal dystrophies do to kind of, look after their sight?
Well, it depends again on the condition. So we know that, for example, smoking is really, really harmful to the eyes. It produces a lot of really nasty chemicals that can actually kill off some of the cells in your eyes. So, people with inherited retinal dystrophy should definitely not smoke. I mean, no one should really smoke with a condition that it can affect your eyes. Other things like, making sure you have a good diet, making sure you're getting enough of your leafy greens because they're really important for the chemicals in your eyes. Also avoiding sunlight because the sun is actually quite harmful to not just your skin, but also your eyes. So if there is inherited disease in your family, then, you should make it a routine to wear sunglasses when it's too bright, stuff like that. But again, like most of the time, if you have an inherited retinopathy, I mean, regular checkups and also making sure that you receive your genetic diagnosis, because I think that is really a crucial element to having any type of inherited disease, is to have a confirmed genetic diagnosis of it.
Excellent, thank you. So we have a question here. So it says, does vitamin A palmitate help RP cells?
Help RP. So it can, I'm sure it can. Vitamin A is really important for even healthy cells. So I imagine it could help. I'm not sure what palmitate is, but vitamin A as a vitamin is very helpful to all retinal cells. And that's because the process of vision actually uses vitamin A. So vitamin A is this molecule that actually can't be made by our body. Our bodies are usually quite good at making the things that we need to survive, but vitamin A is just one that we actually need to obtain from our diet. So you can obtain vitamin A from your leafy greens, like I said, but you can also supplement this. So in some cases, it's really useful. But in some cases, you also need to avoid it. So you do need to be a little bit careful, and you should always consult your doctor first before taking anything like that.
Excellent advice, thank you. So you mentioned earlier about it being really important that people get a genetic diagnosis so they can really understand the condition, the gene that's causing the effect. How can people go about obtaining their genetic diagnosis?
Well, usually if you do have any issues with your sight, you would go to the opticians, and they're likely to refer you on if they think that it's something that could be inherited, if there's family history and stuff like that. So really, if you do notice something with your eyes, the most important thing is to, not leave it. You should go and, seek advice from an ophthalmologist. They'll be able to give you more sound advice, whether it's something that's corrected by glasses or whether it's something that you should maybe investigate a little bit further. I think also your GP would help you with information like this as well, and will refer you in the right direction. But it is important to just not leave it, because ultimately, like I said, if there is a therapy available, we want to be able to get in there as early as possible before the cells die off. So it is really important to go to the doctor and just try and get it done soon as.
That's perfect. And there is more information around getting genetic testing, information about your genetic diagnosis on the Retina UK website. So it's retinauk.org.uk/genetics. So our unlocked genetic package is there. So we've got a couple of other questions. Do you share information with other researchers, either in the UK or worldwide, so that you can learn from their research and vice versa? Excellent presentation and information. Thank you.
Thank you. Yeah, of course we do. Yeah, we try and make research as accessible as possible, not just to other researchers in the world, but also people who live with conditions. And I guess this is like one of the reasons why we do these types of webinars. But yeah, a lot of scientific research, the aim of scientific research is to publish this in journals so that other people can see our work, and hopefully our work will inform the next line of questions that want to be addressed. We also have like large conferences. So I'm actually going to a conference this Saturday. I'm going to the US, so I'll present some of the results that I've had from my studies, and my other colleagues will also present the results that they've had on their studies. And other people from other groups will be there doing the exact same thing. So we do try and meet with other retinal researchers as often as possible to try and find out what's happening in the field, and to keep everything quite relevant, and to make sure that not everyone's doing the same thing. Ultimately, we want to be able to, address as many questions as possible, and we don't want any, redundancy in the research. So,
brilliant, thank you. So Debbie's asked a question. So thank you for the clear explanation and for all your hard work. Do you know anything about the research into RPDR?
Okay, I don't know. So usually in labs, they'll not only study one particular condition, but they'll also study one particular gene, because like I said, many different genes can cause the same type of symptoms and the same condition. So RPDR is a protein, so ultimately the gene, this gene would be quite a large gene, and it needs to have its own line of research, which unfortunately we don't cover in our group. But I'm sure there's much information online, and if I do come across any research groups, I can maybe pass on some information if you're interested in finding out some stuff about that.
That's great. Debbie, I'm sure we have some information within the organisation, so I'm quite happy to share that with you privately afterwards. Rod has asked, have you got any info on cone dystrophy research?
Not particularly. So cone dystrophy, again, it's just where the cones degenerate. So these are these cells that are really important for reading and writing, and they perceive color vision. So some of the conditions that we would work with will have a cone dystrophy. So cone dystrophy is kind of like more of a process. It just depends which cell type is affected first. So there's cone rod dystrophy, rod cone dystrophy. It really depends on which cell type is affected first. So this process we do sometimes see in some cell lines. So in my own research study, I have a patient line that has a confirmed diagnosis of Stargardt disease, but is also so severe that it's actually borderline cone rod dystrophy. So I do a little bit of research on this, but it wouldn't be the main aim of the project that I have here. So I wouldn't really have too many answers for it. But again, I'm sure there's some information on the Retina UK website about this condition.
So with that particular question in mind, would that very much depend on a genetic diagnosis again as to what research is being done about that particular gene?
A lot of the time, yeah. Like we often find that like that's quite a severe or quite a severe symptom to have. So we try and make correlations between mutations and their effects on these proteins that are important for vision. So sometimes a mutation might just, change one very small thing that'll, maybe the protein won't function as well as it used to. Maybe it's a little bit slower at doing something. But for other mutations, they can completely wipe out the function of a protein. And we see this a lot in cone rod dystrophy or cone dystrophy, where basically the protein that's important for that cell's health and survival is just completely lost, and it really accelerates that type of symptom. So you see quite an early onset of disease with that. So again, it is very mutation specific. Yeah, pretty much.
Great job, thank you. So recent news reports said the government had allocated £50 million to help research into motor neuron disease. Does retinal research receive any such government funding? And if not, do you know why not? There's a question.
Yeah, it's a great question, actually. There is a lot of funding available for retina research, and usually in terms of the government, they wouldn't really put out a call specifically for retina research. They would usually just put out a call for general funding, and then you would apply, your lab would apply for that funding and say what it's for. So Linda Lako, who's the leader of this group, that's pretty much her main job, is, you know, searching for calls for funding and being able to provide these applications for funding to try and get that. But that's not just from the government, that's from many different funding bodies in the UK, and especially charity-led ones as well. So like Retina UK fund a lot of projects, and Foundation Fighting Blindness in several different places also do it. So really, we get funding wherever we can, but a lot of funding is actually generated by charities, so it's important to acknowledge that as well.
Excellent, thank you for that, Avril. That leads me very nicely into my little bit at the end. So that is all of our questions that we've had come in this evening. Thank you for being so candid with your answers, and for such a great presentation. It's really good to know that this kind of research is ongoing, and you know, some of the innovative ways that we're looking to kind of people like yourself are looking to find cures and treatments and things for our ranges of conditions. So huge thank you, Avril. Fantastic presentation. So as mentioned at the beginning of this evening's session, we are planning to do at least one webinar every month. And over the next coming months, we have different topics, including different types of technology. We have one around the Access to Work scheme, and we've also got a mini series on Usher syndrome. So those are coming up over the next couple of months. So have a look on our website and our newsletters and things that come out to you for information on those. So Retina UK is a registered charity. We receive no government funding and rely on our wonderful supporters to raise funds needed to provide vital services. Let me try that one again. So we are a registered charity. We receive no government funding and rely on our wonderful supporters, such as yourselves, to raise the funds needed to provide vital services and invest in the groundbreaking medical research, such as what Avril is doing at the moment. So you can now register to walk, jog, or run an ultra marathon for Retina UK for just £1. So there are events going on from the South Coast all the way up to the Lake District. You can register by the 30th of April, so you've got 2 days to do this to get a discounted rate. You can visit www.retinauk.org.uk/trek, or you can call Simon, who is one of our fundraising managers, on 07736 925 174 to find out a little bit more. We'll put this information in a follow-up email. Alternatively, if you'd just like to make a donation to support this kind of work that Avril's doing through Retina UK, we would be so very, very grateful. And you can visit our website. So again, retinauk.org.uk/donate. Or you could even fundraise for us just with your colleagues, or ask your employer to select Retina UK as their organisation's charity of the year. Again, contact the Retina UK fundraising team for more information on how you can do that. So we will be sending out an email over the next couple of days, which has details of where you can re-watch this fantastic presentation, and details on how you can book onto other events that we have coming up. We'll also be seeking your feedback on today's session. We really do value all of your feedback, and yours today will certainly help develop our webinars and our other services going forwards. So Avril, thank you ever so much for your time this evening, and to everybody that's watched tonight, thank you very much and goodbye.